When you get sick, you might wonder, who did I get sick from? And you start going through your social network, your friend groups, roommates, that person on the bus who sneezed on you. But you probably never asked the inverse question. Who did I get that beneficial bacteria from? You see, all over our bodies, we have trillions of microbes that form communities called microbiomes. I study the gut microbiome, which is made up of all of the microbes that live in your gastrointestinal tract. These gut microbes have amazing health benefits. They help you regulate your immune system and perform pathogen defense. We first get our gut microbes when we're born, and they're continuously shaped throughout our lives, with emerging research suggesting that our social networks may play an important role in this. And while we usually think large social network means increased chance of getting sick, it could be that the social transmission of beneficial bacteria counteracts this risk. To investigate this, I must first answer, do social groups predict variation in the gut microbiome? To do this, I turn to the Renzori and Galen colobus monkey. Like humans, these monkeys are primates. We are essentially evolutionary cousins. And like humans, they have a complex social system in which tight-knit groups cluster together to form larger communities. Within these groups, they also have collective care for their infants. These extremely rare behavioral traits are shared with humans and makes this species a useful model to examine how the microbiome and sociality interact. For my dissertation, I'm studying 150 individually identified monkeys that live in Uganda. While in the field, I will collect monkey poop as a proxy for the gut microbiome and later extract DNA for all of the bacteria found in each sample. Last summer, I visited my site for the first time, finding that I could even collect samples from infants. This allows me to look at how social environment also affects early life microbial development. And later this year, I will return to the field for 6 months in order to collect the bulk of my dissertation data. For my analysis, I will connect social networks and microbial diversity together in order to answer my initial question. If social groups do predict variation in the gut microbiome, I can ask finer-grained questions, like how does the strength of relationship affect this? And do infants with more caretakers have a more diverse gut microbiome? The answers to my questions have important implications for how group living affects health. My study system will effectively model how our microbes and our sociality are interconnected in our present and throughout our evolution. Thank you.